KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, so in this video we are discussing phases of cell cycle. Emma, so in this video we are discussing phases of cell cycle, our knowledge point of view. So suppose in a cell cycle and cell division chapter, no, so we are discussing, no, there definitely before one particular cell undergo for the division, no, at the time definitely there are different types of phases over there. So phases of cell cycle, so in this video we have to discuss. Now a typical eukaryotic cell, suppose if you take one particular eukaryotic cell cycle, it is illustrated by human cells in culture. So suppose if you would like to study about the what eukaryotic cell cycle means, so definitely what is the example we will be taking? So human cells we will be taking in culture. Okay, so example point of view, this particular human cells we will be taking. So here these cells divide. So what are the human cells we will be taking? No, these human cells divide once in approximately every 24 hours. This is neat bit, so very important. Means for studies of or for studying of which one? For studying of so these kind of phases of cell cycle. So general example what we will be taking? Human cells we will be taking as an example. So there, so these cells means what are the human cells we will be taking into the consideration for study of phases of cell cycle. So one point we are understanding, that is so these human cells generally divide approximately once in every 24 hours. So thereby, so this particular point you try to save in your mind. Later, however, this duration of a cell cycle can vary from organism to organism. So means here, sorry, human cells we have taken, no, human cells are going to be divided in 24 hours. Likewise, all kind of eukaryotic cells, human cells means eukaryotic cells only, no. So likewise, all kind of eukaryotic cells takes time or takes duration 24 hours for division or for cell cycle means no. So what is the point now we are understanding means, however, this duration of a cell cycle can vary. The duration of cell cycle can vary from organism to organism and also from the type of cell type to cell type. So means here there are different types of factors playing an important role or plays an important role. So thereby here what is the point here we can understand means, so nature of the cell cycle and cell division based on the type of organism or based on the type of the cell, definitely duration time varies. Point you understood now. So that's why here we are studying that human cells will be divided in every 24 hours. So likewise, if I give one more one particular eukaryotic cell example, you will understand. For example, let me take one more example, that is yeast. Let us take yeast for example can progress through this cell cycle so in about 90 minutes. So thereby in yeast cell, so means yeast coming under kingdom fungi, So their yeast cells they definitely will be divided, means a cell cycle and cell division process takes place in 90 minutes, means 1.5 hour. No, so means within a 1.5 hour yeast cells going to be divided. So but here human cells now will be divided in 24 hours. So there, but here what is a common point? Here human cells coming under eukaryotic cells, even yeast cells also comes under eukaryotic cells. The mean, but here human cells are taking 24 hours for dividing a nature for dividing, whereas yeast cells are taking 90 minutes the time. So thereby here one point we are understanding that one based on the cell type and based on the organism, duration of the cell cycle varies. Point purida. So thereby, so here the cell duration or the duration of cell cycle and cell division definitely depends on the cell type, and definitely that will be varies for one organism to another organism. So now cell cycle. So up to here, here two important points we learned. One is human cell is divided once in every 24 hours. Likewise yeast cell that will be divided will be divided every time, means that will divide every once in 90 minutes. Next one, cell cycle is divided into two basic phases. So what is a cell cycle? Here we will be taking now that particular cell cycle will be divided into two basic phases. So first one is interphase and second one is M phase. Okay, so interphase and one more is M phase. M phase is nothing but mitosis. Okay, so now here in M phase point of view, M phase represents the phase when the actual cell division or the mitosis occurs. So means if you take M phase, so what is the indication or what are the processes which occurs in the M phase means? So here in M phase, no, actual cell division or the mitosis process occurs. And interphase, second one, if you take interphase, no, so it represents the phase which is situated between the two successive phases of M. So here for our convenience to study, what is the thing we have divided? Cell division we have divided into two basic processes or two basic phases. So first phase is interphase and second one is M phase. If M phase means, so actual cell cycle and cell division, so actual cell cycle and cell division we will be considering under M phase, whereas interphase, what is it corresponds? So the phase which is situated between the two successive M phases. So that's why here between the two M phases we can find out the interphase, and between the two interphases we can find out the M phase. Okay, so that particular information I will give you in this video, how we have to consider. Suppose if they ask any questions in NEET examinations, how we have to give answer. So wait for 10 minutes, I will explain that side. Next one, now here what is information? Now it is significant to note that in the 24 hours, in a 24-hour average duration of a cell cycle of a human cell, cell division proper lasts for only about 1 hour or only about an hour. So thereby here what is the point here we are understanding means, for example, again we have taken, for example, one more time we have taken the example of human cells there. So one particular human cell is there means that will be divided, now only we learn now that will be divided in 24 hours there. So in 24 hours of duration, cell cycle of a human cell, okay, cell division properly lasts only about an hour. Means for the division of one particular cell, it takes only 1 hour time only out of 24 hours. So thereby, sorry, 24 hours time we have taken. But here cell division point of view, here we are learning only 1 hour. Within 1 hour, suppose if the cell is going to be divided, then 23 hours, what is the function or what are the activities takes place, sir, within the cell means? So here interphase process takes place. So interphase lasts lasts means prolong. Okay, so lasts more than 95% of the duration of the cell. Means now here we have learned cell cycle, definitely our knowledge point of view divided into two basic phases. So here suppose if you take a human cell as an example, for in a human cell, for a completion of interphase, so how long we have to wait? 23 hours. Means 23 hours of duration, definitely one and only the phase of process takes place, that is interphase. Remaining after 23 hours, so 24th hour no, we can observe the mitosis phase or M phase. Amma point purida. So thereby this is an example. Okay, so means interphase, if you consider there in a human cell, it takes or it occupies 95 or it takes 95% of the duration of the cell cycle, whereas if you take a M phase, just it will be completed within an hour. So within an hour. So likewise, somewhat important point I can give you, so try to save in your mind. Example is important. Next one, M phase. M phase starts with, one more time you see there, M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis. So here, first of all, in M phase, sir, what is happening? Sir, M phase means how we have to remember means. So M phase we can start or we can take or we can denote that one or we can identify that one as division of nucleus. Division of nucleus only. So our subject point of view we will be considering as karyokinesis. Karyokinesis is nothing but division of nucleus. So that's why division of nucleus only we will be terming by one more type, like that is nuclear division. Suppose if the nucleus is going to be divided, nucleus is going to be divided, sir, means how, sir, means suppose if you remember the previous class information, no, so within the nucleus we have seen the presence of chromosomes, isn't it? So these chromosomes means what are the chromosomes are present in the nucleus, so those chromosomes are divided. So when these chromosomes are divided or equally distributed to the daughter cells, no, that particular process we will be considering as karyokinesis. Amma point purida one more time. M phase is nothing but or M phase starts with nuclear division corresponding to the separation of daughter chromosomes. So here what are the chromosomes are there? So all these chromosomes are going to be divided. So such particular process only we will be considering as karyokinesis. Point purida. So next one, usually this particular process ends with division of cytoplasm. Means here the M phase within the M phase, within the M phase. Now here two different types of processes we are observing. One is karyokinesis and one more is cytokinesis. So in karyokinesis, what are the main important points we are discussing? Definitely in karyokinesis we can observe the division of division of nucleus, whereas in cytokinesis we can observe the division of cytoplasm. So one more time, in a karyokinesis we can observe the division of nucleus, whereas in cytokinesis we can observe the division of cytoplasm. So means both kind of or both types of processes which occurs in M phase, don't forget. So thereby here in further classes, now here one important point I have given now, in further classes in F phase, so instead of giving of this much of information, I will take M phase can be divided into two types. So one is karyokinesis and one more is cytokinesis. Directly I will take. Okay, ma? So means once you see there, now interphase. So I think so here we are finding, here I think so it is appearing. So interphase for our convenience to study divided into three substages, G1, S, G2. So likewise M phase means what is a mitosis stage is there, no, even that also our convenience to study we can divide it into two types. So one is a karyokinesis and one more is cytokinesis. Karyokinesis is nothing but there what is the process can occurs or what is the process takes place in the karyokinesis? division of nucleus or simply nuclear division. Whereas cytokinesis means so division of cytoplasm. Easy only, no? So try to save in your mind. Now interface though, so the interface though called the resting phase, so called the resting phase is the time duration which the cell is properly or preparing for the division. So here what is the point? Interface, though it is called as resting phase, so here what is the thing we have to understand now? Interface generally, interface generally we will be calling as resting phase. Resting phase means, sir, it takes like sleepy mode, sir, means, or it enters into the sleepy mode means, no. Resting phase means there just the cell cycle process taking place, but there is no chance to the division of cells. But what are the metabolic activities which are required for the division of a cell? No, all kind of processes which occurs in the interface. Koncha lengthy sentence I have used, whether you understood or not. One more time I will tell you once you listen carefully. Interface is nothing but resting phase. So either resting phase means, so why we are calling now, in this particular case, there is no chance, means here the cells are not going to be divided. But what are the metabolic activities or what are the processes which are required for the cell cycle and cell division, all are takes place in interface. That's why, so interface, though it is called as resting phase, is the time duration which the cell is preparing for division. Division undergoing both growth, cell growth and DNA replication in an orderly manner. So there definitely in interface, no, what are the processes are there? No, all these processes definitely takes place in a sequential order or in order manner. So in one sequential order, definitely one and only each and every process occurs or takes place. Next one, in interface, the interface, now already 1 minute back I told you, so interface is divided into 3 further phases or 3 stages simply, or 3 phases. So first one is G1, second one G2 and third one G2. So G1, yes, G2 simply. One more time you see there, first phase is G1 phase, second one is S phase, third one is G2 phase. So sir, if G means what, sir, means G we can be termed it as gap. So that's why G1, no, gap 1. S, synthesis. Sir, synthesis of what? Synthesis of DNA. So in G1 phase or some kind of metabolic activities, in S phase some other kind of metabolic activities, and in a G2 phase also, so some kind of metabolic activities occurs. Now here, so thereby what is the point now I am asking to remember is, suppose if you take interface, that particular interface for our convenience to study, we divided into 3 further phases. So once you try to say along with me, first one is G1, second one is S and third one is G2. G1 is nothing but gap 1, S is nothing but synthesis, and G2 is nothing but gap 2. Now, depth information let me explain. So that is, suppose if you take cell cycle, our knowledge point of view, cell cycle can be divided into 2 basic phases we see now. So there one is interphase and one more is M phase we seen. So first of all, suppose if the cell is going to be divided, first of all, what is the process takes place? Interphase, apro M phase, apro again interphase, again M phase, again M phase, apro interphase, interphase apro M phase. Likewise, first of all interphase, later M phase, again later interphase, again M phase. So likewise, IM or interphase metaphase, interphase metaphase, interphase metaphase. Likewise, in one sequential order, this entire process takes place. This entire process takes place. So that's why here, if once you see there, our knowledge point of view, interface is there, no? This particular interface now we have divided into 3 phases, isn't it? இப்போ பார்த்துக்கிங்க interface 3 phases means, so here you can remember G1 as G2. Okay, next one in M phase point of view, karyokinesis, cytokinesis. Again interface means G1 as G2. That's why to give elaborate information on this M phase and interface, I have written like this. Once you see, இப்போ G1 as G2 அப்புறம் M phase, isn't it? அப்புறம் in interface again G1 as G2 அப்புறம் M phase. Again here interface, in interface again G1 as G2, isn't it? So that's why G1 as G2 together we have taken as I stands for interface, and here also here M stands for mitosis or M phase, later I. So thereby here why I am giving this much of like a detailed information means, suppose if I give like this, very easily you can understand. Sir, what you can understand very easily means, so once you see, suppose if we are talking about the G1, so what you can find out, suppose if you take here, so first point, what is the point you can understand, no? Okay, G1 phase is situated between M phase and S phase. S phase is nothing but synthesis phase, no? So likewise you can understand. And if you take a G2, okay, so it is situated between S phase and M phase, isn't it? Suppose if you take S phase, that is situated between G1 and G2. So thereby if you have this kind of diagrammatical explanation, so suppose if you take a G1 which is situated between the M phase and S phase, if you take S1 which is situated between G1 and G2, and if you take any other particular phase, no, based on this particular small diagram we can denote where it is situated between. எம்மா பாயிண்ட் புரியுதா? ஸோ இது எக்ஸ்பிளனேஷன் ஆல்சோ இம்பார்ட்டன்ட், நல்லா சேவ் பண்ணிக்கோங்க. அப்புறம் let us start to discuss one by one. So what are the phases here we have taken now of interface? So everything, each and every point. So let us take or let us learn the somewhat information related to those. So before giving off this information, so one diagrammatical explanation now let me explain for you. Now diagrammatical explanation of the cell cycle. So there, suppose this one cell cycle phase, if you see there, no, so first one cell we have taken, one cell we have taken. So first of all that particular cell undergoes what is the process? Interface. So in interface, so G1 as G2 processes takes place. So G1 as G2 processes takes place. So G1 as G2 process takes place. So after completion of G1 as G2, next process is M phase. So in M phase now only we see, no, M phase, our knowledge point of view, can be divided into two types we see, no. One is a karyokinesis and one more is cytokinesis we see, no. If karyokinesis point of view, again karyokinesis means now only we see, no, division of nucleus we see, no. If division of nucleus or karyokinesis, so again for our convenience to study we divided into 4 types. Don't get confused. Sariya? Ipo one more time let me explain. Ipo interface is there, no? Interface divided into 3 phases. G1 is G2. Next one M phase divided into 2. One is karyokinesis, one is karyokinesis and one more is cytokinesis. Ipo in karyokinesis again 4 steps are there or 4 phases are there. So first one is prophase. P stands for prophase. Through my voice you can get prophase, metaphase, anaphase, telophase. One more time, prophase, metaphase, anaphase, telophase. Sir, ipo prophase, metaphase, anaphase, telophase, where it will come, sir, means which comes under karyokinesis under M phase. One more time, M phase, if you see there, no, there karyokinesis and cytokinesis are there, no. In karyokinesis these 4 phases comes: prophase, metaphase, anaphase, telophase. Next one mele, here you see there C you are finding, so that C stands for the process known as cytokinesis. So karyokinesis process and cytokinesis process together we will be calling as M phase. I hope my dear students you people are understanding this Otherwise, one more time, once you observe, pause the video, and once you concentrate here, in your mind you think about this. What is a cycle? So how this process takes place? Once you think about it, definitely you will notice the point. Now here once you see that one particular cell, parental cell we have taken, after completion of interphase, means from the G1 to G2, ఇది LMA interphase dana. So this entire phase is interphase. After completion of interphase, EM phase. So definitely if you take one parental cell, after completion of interphase, two daughter cells formation we are observing on the board, isn't it? So one parental cell we have taken, after completion of this entire processes, means interphase and EM phase, now two daughter cells formation takes place. Sir, but here, so here somewhat G not phase, means here once you see there G kile 0 is there, G not phase. So this particular G not phase. So in this video I am not going to give you. In the next video definitely I will give you. But here explanation once I will give you once. What is the information? No. Here some cells, means so what are the cells? No, heart cells, heart, H-E-A-R-T, heart is there, no? So there are heart cells, no? So will not be divided all the time like a normal cells. One more time you see there, heart cells, no, metabolically active da, but will not be divided. Okay, when these generally when these cells will be divided, means when the cells or requirement is there, or when new cells requirement is there, means then only these heart cells will undergo the process of cell cycle and cell division. Other than that, these cells will be in active mode only, means metabolically active da, but will not undergo the process of cell cycle and cell division. Means once the cells or new cells added or new cells are needed, means then only heart cells undergo the process of cell cycle and cell division, and finally new cells are going to be added to the heart cells. Until that, so all what are the existed cells are there, no, will be in G naught phase or quiescent stage or quiescent phase. So in the next video I will mention this explanation. So but here I have given, but one more time I will give you in the next video. Okay, now let us start to discuss G1, S and G2 one by one. Now G1 corresponds correspondence or corresponds to what? It is interval between mitosis and initiation of DNA replication. Now we have started to learn the first one, G1, isn't it? So there here we are here exactly now. So thereby, without seeing the board also, one first point we can write, that is G1, it is situated or it is corresponds to the interval between the mitosis. M means mitosis only, no, mitosis and initiation of DNA replication. DNA replication or DNA synthesis process occurs in S phase only, no. So thereby G1 which is situated between M phase and S phase. So that is a point here we have written. Next point is during G1 phase, sir, what is happening? Means during G1 phase what is happening? No, the cells is metabolically active. The cell is metabolically active. Cell is metabolically active and continuously grows, but does not replicate its DNA. So means here we are studying about the G1 phase. No, in G1 phase definitely cell metabolically very active. So when definitely in G1 phase cell will be metabolically active but continuously grows, means definitely metabolically active means continuously grows in its size. Suppose one particular cell size is there, means that particular cell due to the due to the preparation of proteins and different types of nutrients and different types of metabolically activities, the size of the cell is increased. So that is only we will be terming as, or we will be taking as metabolically active and continuously grows. Definitely the cell size and the cell growth takes place, but there is no chance to the replication of DNA. Okay, replication of DNA process 100% takes place in S phase, isn't it? That's why the replication process which will not occur in G1 phase definitely takes place in S phase. So that particular point only now let us see. Now S phase, now we have started to learn the second one, S1. Yes, S phase or synthesis phase, makes the period during which DNA synthesis or replication takes place. Simple point da. In S phase or in a synthesis phase, what is the point here we can take means, so DNA replication process takes place. Amma, what is the process takes place? DNA replication process takes place in S phase. Okay, so for example, let me take or let me give one example. What is the example? No, suppose initially if you take a total amount of DNA as a 2C. Amma, one more time listen carefully. So if the initial amount of DNA we have taken or it is denoted as 2C, then it is increases up to 4C. Why? Because in S phase now only we see, no, what is the point we see in S is nothing but a synthesis phase. In a synthesis phase, what is happening? Duplication or the replication or synthesis of DNA. Amma, 3 terms I have used right now. Synthesis or replication or duplication. So likewise 3 terms but carries same meaning da. So thereby if the initial amount of DNA is denoted as 2C, if denoted as 2C, so then it increases to 4C. So thereby what is the point we can understand? The DNA, what is the DNA is present in the particular cell? No, that is doubled. Amma, here remember பண்ணிக்கோங்க, DNA மட்டும் double ஆயிடுச்சு. Okay, but however, once you see, however, there is no increase in the chromosomal number. There is no chance to the increasing of chromosomal number. Chromosomal number is a constant only, but DNA content mattum doubled. So 2C amount of DNA is doubled after completion of S phase. Point purida. So next one, if the cell had like a diploid or 2n number of chromosomes at a G1. Suppose so here in G1 2C chromosomes are there, means after S or in S after G2 or in G2 also chromosome number will be 2C only or 2n only. Amma, point purida. Ipo in G1 yes, G2 no, chromosomal number if you take as 2n in all terms, means in G1 2n only, in S 2n only, in G2 also 2n only will be there. But DNA replication point of view, if you consider, once you see there exactly you see at spotlight DNA point of view, mind protocol, don't forget DNA point of view. If you take DNA initially as 2C, where in G1, in S no 4C, in G2 4C da. Why? Because here in S phase DNA is doubled, that's why same content of DNA is moving to the G2. So thereby in if you take a DNA point of view, so DNA point of view in at G1 2C is there, means in S phase no DNA will be synthesized doubled. So thereby here 4C and in G2 also 4C content of DNA only is present. So tomorrow one table also I will give you, so thereby you will understand very clearly. Okay ma, next one. Up to here it is completed. Now next one, even after S phase a number of chromosomes remains as the same as, that is 2n. Means so thereby chromosomal number will not be divided or will not be increased, just DNA number only is doubled or DNA content only is doubled. Now one particular point, one more important point, what is the point we have to be discussed? No, in animal cells during S phase, in animal cells during S phase of DNA replication, during S phase means DNA replication no taking place. During DNA replication begins in the nucleus, whereas centriole duplication occurs in the cytoplasm. Point puriyadama, one more time. If you take animal cells, no, nucleus is there and cytoplasm also, two components are there, no, contents. So in nucleus, no, we can observe the DNA replication in the nucleus, where in animal cells, and in animal cells cytoplasm, no, while DNA duplication process is taking place, so in cytoplasm what is the thing we can observe? Centrioles duplication. Amma, simple only amma, in animal cells, no, in cytoplasm during DNA replication process, means while going on S phase, while S phase process is going on in a nucleus, no, same time only even in cytoplasm, no, we can observe the centrioles duplication. Amma point nalla save panikonga. Okay, in a centriole so in a centrioles, no, centrioles duplication process takes place in a cytoplasm. So while when DNA replication process takes place in a nucleus of animal cells, means here S phase is there, no, in S phase DNA replication process takes place in the same time only in a cytoplasm, no, centrioles process or centrioles duplication also process takes place. So this is a small information regarding G1. Yes, G2 not given still. Okay, so G2 and remaining stages I will give you in next video. Thank you and be safe at home.